Hello friends, today we are going to study about nucleic acid part 3 and in this video we'll study about DNA structure and we'll understand the importance of major groove. DNA is a double helix. Let's first understand with the help of this diagram. Now what all we can see from this diagram? First that two strands are there. This is one strand and this one is another strand. So we know that DNA is a double helix. What else we can see? This is 3' end. This is 5' end. When 3' end facing 5' end is there, that means the strands are antiparallel. These are helically coiled. Now the pairing is there, that is hydrogen bond. In the last videos, we studied that A pairs with T with double bond, G pairs with C with triple bond. And this pairing is known as Watson-Crick pairing. Why? Because this is discovered by Watson and Crick. So this pairing is called Watson-Crick pairing. So wherever in one strand A will be there, in the complementary strand T will be there. If in one strand G is there, then in the another strand it will be C. If C is one strand, then G will be in another strand. If T in one strand, then A will be in another strand. So that means this AGCT and in this strand TCGA, these are complementary with each other. So both the strands are complementary with each other. Now what all we can see? Diameter is 20 angstrom. One helical turn, here it is written, it has 10.5 base pair, which is about 34 angstrom in length. Because the sugar phosphate is not present in a straight line in a 180 degree, therefore they form an angle. This is a small angle, this is a wide angle. So the small angle is known as minor groove and the wide angle is known as major groove. Now let's see about those who discovered this thing. So first, Franklin and Wilkins in early 1950s, they performed the X-ray diffraction studies of DNA and they found that DNA is helically coiled. Then come Watson and Crick in 1953. They said that this double helix is a right-handed coiled structure. These are antiparallel, the chains are complementary, and the pairing is Watson-Crick pairing. Now, one more scientist, that is Chargaff. Before Watson and Crick, the Chargaff was the scientist who in 1940s gave some postulates, and according to his postulates, the base composition of DNA, it varies from one species to another species, but that composition will remain same in all the tissues of the same organism. And this composition does not change with the change in age or nutritional state or the environment of the organism. Now he only said the number of adenine is always equal to the number of thymine present in a DNA. Similarly, the number of guanine is equal to the number of cytosine present in a DNA. He also said, according to this rule, that is A will be always equal to T and G will be always equal to C. That means A plus T will always be equal to G plus C, because number of adenine will always be equal to number of thymine in a DNA and number of guanine will always be equal to number of cytosine in a DNA. This postulates were postulated by Chargaff before Watson and Crick double helical model. Now base flipping. What do we mean by base flipping? In a DNA, the strands are bonded with each other. These are bonded with each other. Now base flipping means whenever any protein comes to interact with the DNA, for example, repair enzymes, recombination enzymes. So when these proteins come to function in a DNA, so they should interact with the DNA. So that means these proteins or enzymes, the amino acid side chain of these enzymes should interact with the DNA. Then only they can perform repair or recombination. So for that, it is not like that always the full DNA will be opened up. What it will do, it will flip. For example, if suppose C should bind in the catalytic site of this particular enzyme. So like this, one base will be flipped out so that they can fit in the catalytic site of the enzyme. When the function is done, it will again return to its original place, forming the bond with the complementary strand. So this is known as base flipping. So base flipping is done so that Without opening the DNA double helix or without disturbing the double helix, the enzyme can perform their function. So this phenomenon is known as base flipping. Now DNA double helix has minor and major grooves. So as in the last slide we studied, this is a minor groove. This side is major groove. So minor groove is approximately of 120 degree. Major groove is approximately of 240 degree. This grooves or this angle developed because the sugar phosphate backbone is not in a straight line. That is at an angle of 180 degree with each other. So because the backbone is not in a straight line, therefore these major and the minor grooves form. Now this major groove is the part that store the information. The information of the DNA is stored in this major groove. How? Let's see. Major groove is rich in chemical information. In any groove, whether it is a major or minor groove, the bases which are involved in bond formation, they will expose the groups that are involved in bond formation like hydrogen bond donor, acceptor, the hydrophobic site. So here, the edges of each base pair, these are exposed in the grooves, which create a pattern. The patterns of hydrogen bond donor, acceptor, hydrophobic group. Okay, so they will create a pattern that forms a code. How? We'll understand now. So now here, as you can see, when G is bonded with C, this is first figure, second, third, and fourth. So in the first figure, when G is bonded with C, the upper side is showing the major group. The lower side is showing the minor group. In this pairing, in the major group, the sites which are exposed are A, D, and H. That means A for hydrogen bond acceptor, D for donor, hydrogen bond donor, H for nonpolar hydrogen, and M for methyl groups. So here in the major groove, as we can see that when G is paired with C, so the sites that are exposed are two acceptors, one donor, and one nonpolar hydrogen, forming the code AADH. So here I'm writing AADH is for when G pairs with C. But in the minor groove, if we see, the sites that are exposed are ADA. This is for major groove. For minor groove, the sites exposed are ADA for GC pairing. Similarly, when C pairs with G, in case of major groove, the sites that are exposed form the code HDAA for C pairs with G. Similarly, in the minor groove, it is ADA for C and G pairing. Now with this only, we can understand that there is a difference. From the major groove, we can easily understand whether G pairs with C or C pairs with G. That is, with G pairs with C, it is AADH. When C pairs with G, it is HDAA. So this can be differentiated. But in the minor groove, if you see, then in both the cases, whether it is G pair with C or it is C pair with G, it is ADA. So in the minor group, it is very difficult to differentiate the pairs. Similarly, for A and T also, in the major group, in this third figure you can see, it is forming ADAM, whereas in minor group it is AHA. Similarly, when T pairs with A, it is MADA, whereas in minor group, again it is AHA. So in the minor group, it is very difficult to differentiate the pairs, whereas with the help of major group, it is easy to differentiate the pairs. So that's why major group is rich in chemical information. Major group gives the information that in the DNA, whether A pairs with T, T pairs with A, G pairs with C, or C pairs with G. So this information is used by proteins that interact with the DNA. How come a protein know that in DNA sequence where to bind without disturbing the double helix? So this is the codes, that is the major groove code that helps to identify the DNA sequence by the protein so that it can bind at the right place. So major groove, it gives the precise chemical information. So the pattern is important because it helps protein to identify DNA sequences without opening it. So now one question from my end for you all, that is, explain what is meant by the following adjectives assigned to double-stranded DNA or the two strands that make up the DNA. Polar, antiparallel, complementary. What do you understand by these objectives? Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.